Saccharomyces cerevisiae is a model organism. It is a yeast cell that has a cell membrane and a cell wall. Saccharomyces cerevisiae encodes for the protein enhancer of Gal4 DNA binding, or EGD2. EGD2 forms a heterodimer with a similar protein called EGD1 to form the nascent polypeptide-associated complex, or NAC. NAC is associated with cytosolic ribosomes that translate mRNA into polypeptides. During translation, NAC associates with the newly synthesized polypeptide chain emerging from the ribosome. It allows translocation or transport of the growing chain from the cytoplasm of the cell to the mitochondria or the endoplasmic reticulum. Proteins translocated to the endoplasmic reticulum can therefore be modified and transported all around the cell to perform their function. For example, ergosterol is a key component of the fungal cell membrane that provides fluidity and controls permeability of the cell. It is synthesized by various enzymes in the ergosterol biosynthesis pathway. The antifungal fluconazole inhibits the enzyme cytochrome 14 alpha lanosterol demethylase that converts lanosterol to ergosterol. Therefore, in the presence of fluconazole, there is a reduction in ergosterol production. Additionally, ergosterol in the cell membrane that is replaced by toxic 14 alpha methylated sterols. This causes an increase in flexibility and permeability of the cell. That causes an influx of reactive oxygen species that result in increased oxidative stress in the cell. This can cause protein misfolding and cellular death. It was noticed that in the presence of fluconazole, EGD2 expression is downregulated. But why? We propose that the downregulation of EGD2 is to reduce the ability of fluconazole to target P450-14DM by slowing the rate of translation and therefore to minimize the downstream effects of the integration of the 14 alpha methylated sterols in the cellular membrane, increased membrane permeability, and increased oxidative stress on the cell. Therefore, we hypothesize that if this is true, then a strain with knockout EGD2 would grow better than wild type in the presence of fluconazole. However, if knockout EGD2 strains were exposed to cell membranes and oxidative stresses that mirror the action of fluconazole on the cell, it would not confer an advantage and wild type would grow better. So to test this, we generated a dilution series of concentrations from 10 to the 6 to 10 to the 1 cells per mil, wild type and EGD2 knockout mutants in PBS that were spot plated onto the following conditions: no stress, negative control, fluconazole at 16 micrograms per mil, oxidative stress, cell membrane stress, and a combination of cell membrane with oxidative stress to examine if there was an additive effect, and were incubated at 30 degrees Celsius over a 1-week period. It was found that there was no difference in the growth between the wild type and knockout strains in the no stress conditions. This indicates that the deletion of EGD2 does not result in underlying growth differences, and that the growth can be directly compared in the stressing conditions. When we examined the fluconazole plates, there was not much difference in the growth of wild type and knockout strains. It appears that there was a slight increase in the ability of the knockout to grow as hypothesized. However, under the presence of the stresses of oxidative stress or cell membrane stress individually, there was no difference in the ability for knockout and wild type strains to grow. Perhaps the stressing concentrations were not high enough to observe a change in the phenotype. As fluconazole targets both the cell membrane and increases oxidative stress in the cell, we looked at a combination treatments to mirror the effects of fluconazole. It was found that knockout was able to grow better than wild type strains. This was unexpected because of EGD2's main function was to prevent the incorporation of toxic sterols into the cell membrane, that these stresses would result in a decrease in growth in comparison to wild type. This result therefore suggests that instead of EGD2 being downregulated in response to fluconazole, so it can reduce transcriptional or translational activity, so that cell can determine changes in gene expression so that it can cope with the stress better. Therefore, this allows the knockout strain to go better in the long run in these stressing conditions than compared with wild type. This is supported by the findings of Hiroyuki et al. In 2009, that found that EGD2 is downregulated in response to stressful conditions by ubiquitin ligase RSP5. So, this study has shown that the downregulation of EGD2 in response to fluconazole is not directly related to antifungal resistance, but is instead a part of the mechanism of Saccharomyces to deal with stress in general. For future experimentation, a protein assay would be performed to confirm that the EGD2 protein is being downregulated in the wild-type strains, and increasing the range of concentrations of the treatments may improve our understanding of how they affect the cell growth. Some of the treatments did not seem significantly different from the wild-type in terms of colony growth, but additional replicates may provide a more accurate representation of growth patterns in response to each treatment. Having additional replicate plates would reduce the probability of any variation in growth being attributed to error or chance. As EGD2 protein is a part of a protein complex, further study could include knocking out its other subunits to compare those effects on the growth with those of this study. This may lead to a better understanding of how to improve antifungal treatment in conjunction with fluconazole. For instance, Raymond et al. In 1999 showed that when genes for the other subunits are deactivated, the EGD2 subunit on its own can become toxic to the fungal cell when not associated with its other subunits, such as EGD1.